When we think of omega-3s, we usually think of heart health. But EPA and DHA, the omega-3s from fish, also help your body burn fat and build muscle. For fat burning, EPA and DHA really rev up mitochondria, your fat-burning engines. This helps your cells burn more fat, make more mitochondria, and even waste a little extra energy as heat instead of storing it. For muscle, omega-3s make your muscles more responsive to protein. So after a high-protein meal or a workout, your muscles repair and grow more effectively. Here's the key. Plant omega-3s, like those from flax or chia, don't provide these benefits. Your body barely converts them. You need EPA and DHA from fatty fish or fish oil supplements. If you want better fat burning, better recovery, and better muscle health, omega-3s are a simple, evidence-based choice. This is lecture 128 of the Metabolic Classroom. Looking to improve your own metabolic health? Visit insuliniq.com for courses, coaching, consultations, and a 10-day free community membership trial. To dive deep into the science behind metabolic health, become an insider at benbikman.com, where you'll enjoy my exclusive content, ad-free podcasts, live stream Q&A access, and more. Welcome to another mini lecture in the Metabolic Classroom. I'm Ben Bikman, a biomedical scientist and professor of cell biology. Today, we're diving into omega-3 fatty acids and their remarkable effects on both fat metabolism and muscle growth. Over the years, you've most certainly heard about omega-3s, likely in the realm of heart health or inflammation. But what you might not know is that these essential fatty acids have profound effects on how your body burns fat and builds muscle. Before we get into their metabolic effects, let's establish what omega-3 fatty acids actually are. Omega-3s are a class of polyunsaturated fatty acids characterized by the position of their first double bond. So 3 carbons from the omega, or the methyl end of the molecule. That's the omega-3. There are 3 main omega-3 fatty acids we care about metabolically. First is alpha-linolenic acid, or ALA. This is the plant-derived omega-3 found in foods like flax seeds, certain nuts, and chia seeds. ALA is the shortest of the omega-3s at 18 carbons, and while it has some reported health benefits, it's not the most metabolically active, certainly not in the context we're discussing. So of the 3, it is the least relevant in this particular context. The 2 omega-3s that really matter for fat and muscle metabolism are the longer chain forms, and they are EPA, which stands for eicosapentaenoic acid, which has 20 carbons and 5 double bonds, and then there is DHA, or docosahexaenoic acid. That's a 22 carbon with 6 double bonds. And here's something critical to understand. EPA and DHA are found in animal foods. The richest sources are fatty fish like salmon, mackerel, and sardines, but you'll also find EPA and DHA in more modest amounts, albeit, but in other animal fats that are more commonly consumed, certainly in the Western world, like beef, lamb, pork, poultry, eggs, and dairy. Any animal fat will contain some degree of EPA and DHA, the 2 omega-3s that you need. The amounts will vary based on what the animals ate, with pasture-raised and grass-fed animals generally providing a little more, but they are modest differences. Fish are so rich in EPA and DHA because they accumulate it from algae through the marine food chain. And here is a key point. You will not find meaningful amounts of EPA and DHA in plant foods. Plants make ALA, not the longer chain forms your body needs for the metabolic effects that we're going to be discussing today. Now, you might be wondering about ALA from plants. Can't your body just convert that plant-based omega-3 into EPA and DHA? Because there are animals that can. So technically, yes, but practically, it's not feasible. The conversion is so inefficient. This is just in the order of a few percentage points as the degree to which ALA can convert to EPA or DHA, that ALA is essentially metabolically useless for the fat burning and the muscle building effects that we're discussing here. So for all practical purposes, and we'll revisit this at the end, If you want the metabolic benefits of omega-3s, you really need to be focusing on EPA and DHA directly from animal sources. Plant-based omega-3s simply will not deliver these effects. Now, let's talk about how omega-3s help our bodies burn fat. The effects here are fascinating and work through multiple mechanisms, but they all converge on the mitochondria. One of the most important effects of omega-3 fatty acids is their ability to stimulate mitochondrial biogenesis, literally the creation of new mitochondria. When you consume EPA and DHA, studies show they upregulate key transcriptional factors involved in mitochondrial biogenesis, in particular PGC1 alpha and NRF1. PGC1 alpha is often called the master regulator of mitochondrial biogenesis, and omega-3s increase its expression in skeletal muscle and, importantly, adipose tissue. So it's even stimulating more mitochondria in your fat cells, which you would traditionally think are just there to store energy. But if the mitochondria are there, it certainly suggests we are burning more of it as well, because more mitochondria means a greater capacity for fat oxidation or fat burning. Your cells literally become better fat burning machines. Beyond just making more mitochondria, omega-3s also make the mitochondria you already have work better at burning fat. They do this by increasing the expression and activity of carnitine palmitoyl transferase 1, or CPT1. This is the rate-limiting enzyme that shuttles long-chain fatty acids into the mitochondria for beta oxidation. Remember, beta oxidation just means you're burning the fat. Without CPT1, these long-chain fatty acids, which is the vast majority of all the fats we burn, simply can't enter the mitochondria to be burned for fuel. Research shows that omega-3 supplementation increases CPT1 expression in skeletal muscle, liver, and adipose tissue, or fat tissue. One mechanism appears to be through activation of AMPK, which is essentially the cell's energy sensor. When a cell becomes low with its ATP, or the main energetic molecule, AMPK will turn on, and essentially it just ramps up any effort to increase ATP production. So it's really increasing the burning of any fuel. When EPA activates AMPK, it triggers a cascade that increases CPT1 expression and fatty acid oxidation. This creates an environment where your body preferentially burns fat for fuel rather than storing it. Now, here's where things get really interesting. Omega-3s also promote mitochondrial uncoupling, particularly through upregulation of uncoupling proteins like UCP1 in brown adipose tissue and UCP3 in skeletal muscle. Let me explain what uncoupling means, although I've certainly elaborated this point in previous mini lectures in the metabolic classroom. Normally, when mitochondria burn fuel, they create a proton gradient that drives ATP production. That is, in its simplest, what we could call coupled respiration. But uncoupling proteins allow protons to leak back across the mitochondrial membrane without generating ATP. Now, I understand that is a little complicated, although accurate, but let me explain this again with a brief analogy. Coupled mitochondria are like you driving a car when the car is in gear or it's in drive. If you press the accelerator, you'll see the RPMs rise, reflecting that the engine is burning fuel, and you will see the speed increase. You're moving faster. You're getting more work for the amount of fuel you're burning. But uncoupled mitochondria are like you revving the engine while it is still in park or neutral. The engine is burning fuel and you see the RPMs going up, but you're not getting any work or action from it. This might sound wasteful, and it is, but it is actually metabolically beneficial. Back to the mitochondria in the human body here, because it forces your mitochondria to burn more fuel to maintain their function, and that extra energy is released as heat rather than being stored as fat or having to be burned through the process of exercise or physical activity. Research in rodent models has shown that omega-3 supplementation significantly upregulates UCP1 expression in even white adipose tissue, effectively promoting browning or beiging of white fat. That is when you're converting the energy-storing white fat into the energy-burning beige fat. So the white fat starts to behave more like brown fat. Studies also show increased expression of UCP3 in skeletal muscle, which can just further enhance the uptake and burning of fat. When you supplement with omega-3s, these fatty acids literally become incorporated into your mitochondrial membranes. So this is an entirely novel approach or aspect of omega-3s in all of this as well. A landmark study in human skeletal muscle found that 12 weeks of fish oil supplementation robustly incorporated EPA and DHA into mitochondrial phospholipids. So they were literally becoming a part of the membrane. And this isn't just some kind of passive incorporation. In so doing, we actively change the mitochondrial function. Specifically, omega-3 enrichment of mitochondrial membranes improved ADP sensitivity. ADP is the precursor molecule that the mitochondria will use it will take that and it will combine one more phosphate onto that and then generate ATP, the main molecule we're interested in when it comes to the mitochondria. The study found that mitochondrial respiration in response to ADP was enhanced, suggesting improved coupling between energy demand and ATP production. Interestingly, they also found increased mitochondrial capacity for ROS emission or reactive oxygen species production, but without any increase in oxidative damage markers, suggesting omega-3's enhanced antioxidant defenses alongside their improved sensitivity to ADP or an enhanced ability to make ATP. The unsaturation of mitochondrial membrane lipids matters tremendously. More unsaturated fats in the membranes, which is what you get with omega-3 incorporation, are positively associated with higher rates of palmitate oxidation. This makes some sense. Fluid flexible membranes allow the protein complexes of the electron transport chain to function more efficiently. Rather than being more rigid in the structure, they are more dynamic. All right, now let's shift from fat to muscle. The effects of omega-3s on skeletal muscle are equally impressive, as far as I'm concerned, but they do work through different mechanisms than the fat-burning effects. It's no surprise. Muscle cells are very different from fat cells. The primary mechanism by which omega-3s support muscle growth is by enhancing muscle protein synthesis, the process by which your muscles build new proteins from amino acids. This is where the research gets particularly compelling. A groundbreaking randomized controlled trial in older adults found that 8 weeks of omega-3 supplementation, providing about 1.8 grams of EPA and 1.5 grams of DHA per day, significantly increased the rate of muscle protein synthesis during a hyperaminoacidemic, hyperinsulinemic clamp. So that's just basically a state where both amino acids and insulin are elevated. The key finding was that omega-3s didn't increase basal muscle protein synthesis during fasting, but they dramatically enhanced the anabolic response to amino acids and insulin, increasing protein synthesis rates by more than 3-fold compared to pre-supplementation values. This same effect has been replicated in younger adults. Another study found that omega-3 supplementation augmented the muscle protein synthesis response to both elevated amino acids and insulin in healthy 25 to 45-year-olds, again, without affecting basal rates. What this tells us is that omega-3s don't stimulate muscle growth on their own. They amplify the anabolic signal from nutrients. But how can these fats help grow muscle? The answer lies in one of the most important signaling pathways for muscle growth, the mTOR pathway. mTOR, which stands for mechanistic target of rapamycin, is essentially the master regulator of cell growth and protein synthesis. When omega-3 fatty acids are incorporated into muscle cell membranes, particularly the phospholipids of the sarcolemma and the intracellular organelles, so the sarcolemma being the membrane of the muscle cell, then the intracellular organelles would be things just like the mitochondria, but they alter the membrane structure in ways that enhance the activation of mTOR and its downstream targets. When mTOR is turned on, it will in turn go and turn on many other proteins. And multiple studies have shown that omega-3 supplementation increases the phosphorylation or activation of mTOR. The enhanced protein synthesis translates into real-world gains when it comes to muscle mass and function. A meta-analysis of randomized controlled trials in elderly populations found that omega-3 supplementation led to modest but significant gains in muscle mass, an average of 0.3 kilograms. When the analysis looked specifically at higher doses above 2 grams per day, the benefit doubled. It went up to almost 0.7 kilograms of muscle gain over the length of the study. Remember, this is in older adults, so any gain is impressive. But even more impressive, omega-3 supplementation has been shown to improve muscle strength. In older adults combining omega-3 with resistance training, studies found significantly greater increases in 1 rep max compared to resistance training alone. The mechanisms likely involve both enhanced protein synthesis and improved mitochondrial function that we've discussed, allowing muscles to generate more force and recover more effectively. One of the most clinically relevant effects of omega-3s is their ability to prevent muscle loss during periods of disuse. When a limb is immobilized or during bed rest, muscle rapidly atrophies through increased protein breakdown and decreased protein synthesis. Omega-3 supplementation has been shown to attenuate this loss by maintaining protein synthesis rates and reducing the expression of factors that regulate protein breakdown. This has major implications for aging populations, surgical recovery, and even injury rehabilitation, situations where muscle disuse is unavoidable, but muscle preservation is critical for functional recovery. The common thread in all these muscle effects is, once again, membrane incorporation. When you consume EPA and DHA, they progressively replace other fatty acids in the phospholipids of muscle cell membranes. Studies show this incorporation begins within the first week of supplementation and continues to increase for at least 8 to 12 weeks. This membrane remodeling has profound functional consequences. Enrichment of EPA and DHA into the sarcolemma and the mitochondrial membranes alters membrane fluidity, lipid raft composition, and the function of membrane-bound proteins, including receptors like insulin receptors, transporters like glucose transporters and fatty acid transporters, and even signaling molecules like the one we just got done talking about, mTOR. This is how omega-3s can simultaneously enhance insulin signaling, improve amino acid sensing and transport, facilitate mTOR activation, and optimize mitochondrial respiration. They fundamentally change the operating environment of the muscle cell. So what does all this mean for you? If you're interested in optimizing body composition, namely burning more fat, building more muscle, who isn't? Omega-3 supplementation appears to be a valuable tool. Based on the research, effective doses for metabolic benefits range from about 2 to 4 grams per day of combined EPA and DHA. The studies showing muscle protein synthesis benefits typically around 2 to 3.5 grams daily, while the fat oxidation in mitochondrial biogenesis studies used similar or even slightly higher doses. Now, let me be very clear here. The benefits require EPA and DHA from animal sources. The plant-based omega-3 ALA will not provide these metabolic effects. The conversion rate is simply too poor, and ALA is simply not a part of this process. So if you are relying on flax seeds or other nuts or seeds for your omega-3s, there may be some benefits to that, and I'm not telling you you shouldn't be consuming them, but if anything in the midst of this mini lecture has stood out to you, indeed you now want some of these benefits, you need to focus on animal sources. Of course, fatty fish are going to be the best, and you want to consume them regularly. So if you're supplementing, look for products that specify their EPA and DHA content, not just total fish oil or omega-3s, because you want those long-chain animal-derived forms. And also, timing will matter for the muscle effects. Since omega-3s amplify the anabolic response to amino acids and insulin, it makes sense to ensure you have adequate omega-3 status when you're consuming protein-rich meals or some kind of post-workout period when you're consuming more nutrients. However, the membrane incorporation effects build over weeks, so consistent daily intake is probably going to be more important than precise timing. In closing, the metabolic effects of omega-3 fatty acids extend far beyond their well-known cardiovascular benefits. Through their incorporation into cellular membranes, particularly in mitochondria and muscle cells, EPA and DHA fundamentally alter how your body processes energy. They enhance mitochondrial biogenesis and function, increase fatty acid oxidation, promote metabolic or mitochondrial uncoupling, amplify muscle protein synthesis, and help even preserve muscle mass during periods of stress or disuse. Thanks for joining me in the Metabolic Classroom today. Class dismissed. Remember, more knowledge, better health. Looking to improve your own metabolic health? Visit insuliniq.com for courses, coaching, consultations, and a 10-day free community membership trial. To dive deep into the science behind metabolic health, become an insider at benbikman.com, where you'll enjoy my exclusive content, ad-free podcasts, live stream Q&A access, and more.